everybody. Thanks for joining. So they can hear us online now. Should I project my voice or should I just speak naturally?
Projecting would be great.
Okay, thanks. Well, thanks for inviting us to come speak with you here at CCMB. So there are going to be three of us speaking. So I'm Jeremy in the top there. I think all of you know Dr. Razun and Dr. Mishra, and we will each kind of briefly introduce ourselves as we go through. Here is our disclosure slide. And here is the summary of the entire talk in case you want to tune out, check email, whatever you want to do, or you could listen, your choice. So these are our pillars, right? So we're the Center for Clinical Cancer Informatics and Data Science. And we're built on 5 pillars. Those are content standardization, clinical integration, information extraction, dissemination of new technology, scholarship and education, all within the confines of the cancer space. We'll come back and talk about each of these things a little bit more towards the end. The other important slide here is our contact information. And so I'm the director, Dr. Zun's the associate director, Dr. Mishra is our coordinator. You can reach us at Brown email or Lifespan emails. Take note that Lifespan emails are not checked very often and they're going away in about a month. So maybe don't. These are Brown emails. Yeah, Brown emails also check them more often. We're hoping to have a website up and running real soon. So briefly about me, so I'm Jeremy Warner, like I said. I'm a practicing clinician, and I have a few too many board certifications that I have to maintain. Pretty annoying, but I do practice in hematology as my clinical area. I was at Vanderbilt for about 10 years before coming here. Yeah, come on in. Here's seats. So the main reason I came here is actually to create the center that we're going to talk to you about. I'm also the editor in chief of GCO Clinical Cancer Informatics. It's not a coincidence that there's similarities to the names there. And 2 pictures I just want to show you. So one is just a little bit I like to hike. In fact, I'm looking, we should talk about this. I'm looking to start bagging some peaks up in New Hampshire and Vermont, but I just haven't quite the drive to drive up there. But this is a picture we just took up in the Alps in Austria. And then this thing on the right, does anybody know what that is? It's actually not a cricket, but I thought it was. It's something called a trig. It's called the Colombian trig. And it's a symbol of global warming, actually. It is native to Central and South America and the Southern United States, and it's actually moving up here. And I just saw that on my table. That's my table outside my house last week. So, yes, yeah, it definitely is. But it's just like me coming from the South. Here's another Southern person, creature. Okay, so getting into it, what is clinical cancer informatics? Like, what are we talking about here? And I like to, this is informatics with cancer. So informatics, if you could sum it up in just a few words, is this action or process of taking data, transforming it into information and into knowledge and into wisdom. I'm going to give you some examples of what that means in the cancer space. Have you heard of that thing? So I'm going to get rid of that, turn up the volume. Maybe
you can just close it.
Yeah, no, because it's like the mouse was under it. Don't worry, it's fine. We don't have to worry. That's cool. It's fine. You can all see this, right? Like it might cover up. Actually, it will cover up some stuff. Yeah, do you know how
like we should maybe close the presentation.
There we go. Cool. And then go back to
Under more.
well, I think it's gone, hopefully. Let's get back to the show.
Yeah.
Sweet. All right. All right, so this is that pyramid that I just showed you, looking from above, right? Yes.
We're no longer sharing your screen.
It is sharing. It's over. No, it is. It is sharing. Yeah. Stop. Okay, so let's see. Desktop 2.
Distribution.
Good.
Okay, yep.
Awesome. All right, here we go. All right, so now I'm showing this pyramid as a above view here. And I just want to kind of talk about three aspects of cancer. And those would be phenotype, genotype, which I think CCMB spends a lot of time thinking about working on, and environment. And if I can get this to actually go. There we go. Okay, so at this base level of data, so that's very basic measurements. And so in the phenotype world, that would be something like, what is the histology of the cancer we're looking at? Look under a microscope. In genotype, that would be things like the sequence of the tumor. And then the environment, that would be pollution levels. Now, if we step up a level, we start to create meaning, right? So we're transforming data into information. In the phenotype world, that would be saying something about the behavior of this thing that we see under the microscope. Is it an aggressive invasive cancer, or is it a benign neoplasm that's not going to cause problems? In the genotype world, we're talking about pathogenicity. So these sequences that we've observed, what are they actually, are they pathogenic? Are they actually driving the growth of the cancer? And similarly in the environment, are pollutants pathogenic, or are they just wrapped? Now, if we step up another level to what we call knowledge, now we're getting into very close to the clinic here, right? So in the phenotype, we're talking about not just behavior, not just histology, but what are we going to do? What are the options? What can we do to treat the cancer, for example? Same thing in genotypes. Now we've stepped up to level 2. Are there actions we can take to treat based upon what we've observed in the genotype? And in the environment, are there things we can do? Are there risk modification behaviors that we can undertake? And then kind of going up to that last level, wisdom, which is very elusive for all of us, I think. Again, in these 3 zones, in the phenotype, that might be something about the patient's values and preferences, which may or may not align with the treatment options. In genotype, that might be something like understanding what's going to happen if we treat an observed mutational profile, what's going to happen as evolution occurs under pressure. And then the environment, this might be things around social determinants, social justice, structural racism, other influences on outcomes. So that's one way we think about clinical cancer informatics. The other way is kind of from this research data ecosystem point of view. And so this slide has a number of items on it, but what I want to basically bring it down to are kind of 3 chunks. There's a data collection process, there's a data extraction process, and then there's the creation of data models. It's the stat news article. Okay, so what I want to do now is just briefly talk about kind of each of these areas and illustrate with some projects that we work on currently. So data collection. So I mentioned I came from Vanderbilt, and you may or may not know that REDCap also comes from Vanderbilt. So is everybody who's not used REDCap? Raise your hand if you've never used REDCap. All right. And I guess the rest of you have, right? So okay, raise your hand if you have used REDCap. It's actually smaller than I figured. Okay. All right. So REDCap's a really cool data collection tool, which actually has, in this slide now is probably 4 or 5 years out of date, but it's got over 6,000 institutions, over 2 million users, and over 23,000 articles that have used REDCap in one way or another. Why am I mentioning REDCap? Because it really drove what became our COVID-19 and Cancer Consortium, which is one of our first big projects. So back in 2020, March, we created this consortium, and you can see here the mission statement, which is to collect and disseminate prospective, granular, uniformly organized information on people with cancer who are diagnosed with COVID at scale and rapidly as possible. This is our data models completely in REDCap, so we really built this thing around this idea of data collection. I don't have obviously time here to talk about our consortium in detail, but we ended up expanding to over 90 contributing centers. There's some issue with some of the things.
There's some color schemes.
Yeah. Well, anyway, you get the point here. It's that we had a large consortium, over 17,000, actually close to 20,000 cases were collected. Here's a map of our participating sites. We just in over 3 years had 106 sites participate at one point or another. Actually, this is the largest COVID-19 and cancer registry in the world. And these are our contributing sites, a little different oriented here. We've had more than 20 publications, almost 600, more than 600 authors at this point, right? Many citations. We have a website that you can learn about. We have more work to do. So I think maybe a lot of people think COVID's, or want to think COVID's over and done with, but I think there's a lot of unanswered questions which we're currently thinking about, and these are just a few of those. And by the way, this all requires data collection to answer this, right? So that's why it's under this category. Understanding stuff, understanding about long-term symptoms and complications. Does having COVID infection affect the long-term outcomes of people with cancer? Does anti-cancer therapy work differently in the setting of SARS-CoV-2 infection? Are there imaging changes? We know there are imaging changes. This actually interferes with our ability to understand whether someone is progressing or not. Dr. Zoom's going to talk about recurrence later. One of the ways we detect recurrence is looking at lymph nodes. COVID makes lymph nodes big, so there's actually like a very interesting and problematic new phenomenon here, understanding whether someone has progressed to COVID or not. We want to understand more about variants, which are constantly evolving, as we know. And sort of in the different dimension, we want to understand effects on emotional distress and anxiety. COVID increased loneliness in patients with cancer, understanding what that means. And cancer screening, delayed diagnosis. There's a lot of different questions. And just very recently, we received UO1 funding to work on these questions and more. So we're pretty excited to continue that work. Moving on to data extraction. So I'm going to illustrate this kind of domain with something called DeepV. So DeepV is our project, which stands for Cancer Deep Phenotyping. The idea here, have any of you done a chart review? Gone into the medical records ever? All right. I see nods in the back. Yes, actually, you. Right? But you've probably worked with clinical data in your work, right? Like you've worked with some variables that are called clinical. Very few. Okay.
I didn't present it.
All right. Well, so we'll talk about that.
Good IRB.
Yeah. Okay. Yeah. Well, the thing is, like the electronic, it's daunting, right? So, like, if you print it out in electronic health, it would be like that, thousands of pages. And that's mostly why I think more biologically oriented folks stay away from it. It is very complicated and very time consuming. Well, the idea of DeepV is it actually goes in there and gets the information for you, puts it into a computable format, and then you can use it in your analyses. So just to this is going to be way too small for you to see. This is kind of like how it works. It basically digs through all these different aspects of the chart. It can be things like what a clinician is writing, a pathologist, a radiologist, like a brain MRI. And each of these notes has various important features that can be pulled out and structured and turned into a summary for each patient. So this here kind of gets into it a little bit more. I'm not going to spend too much time on the weeds here, but basically DeepV is a multi-modular system that's basically using state-of-the-art methods for each aspect of what it's trying to pull out. This is kind of an example of how it works. A fairly simple sentence there about a breast cancer phenotype. And it's breaking that down, it's labeling various concepts, it's figuring out how they're related to each other. So we know what is means, right? Right? It's pretty clear to us. But a computer doesn't necessarily know what that means, right? Like, so tying that together and understanding that certain findings are negative, negations, and then getting the phenotype. And then this just shows the example again. This is what's in the yellow here and what's in the yellow here are the same piece of information. Right? It's actually the same. Here doesn't know that. So this is the process by which you get to the result, which is a consistent finding. Right, so that's the last step here. So kind of putting it all together. So imagine these come from different documents. This one came from a pathology report on the left. This one came from an emergency room. Note on the right. We kind of do this at scale for the entire record. We've done this in breast cancer, melanoma, ovarian cancer. A limited number. It's actually a difficult process because there's different aspects of different kinds of cancer, but we are working on generalizing. Last thing I want to talk about briefly before turning it over here is about data models. So, we're collecting data, extracting data from records. How do we put that together in a comprehensible way? How do we represent it as knowledge? And this is something I've been working on for years, called the Hemont.org. It's a website, a wiki, exactly as Wikipedia, same idea, same software. And our goal is to create a knowledge base to capture all standard of care systemic anti-cancer treatment. So this would be what you hear as like chemotherapy. What is her name? Kate Princess. You'll probably saw she just finished her treatment. You don't know exactly what it was, but you know it kind of it's a systemic treatment. And so it's important to appreciate that cancer is many things, right? So on this website, we have over 360 subpages for different kinds of cancer, over 1.4 million lines of content, and it's a big complex undertaking. This is just a quick diagram of who uses our website. So about 25 to 30,000 visitors a month, primarily healthcare professionals, practicing folks, but also researchers. There's other use cases. We have partners that you can see below that we work with. You can see that most of our visitors come from the US, but we have had visitors basically from every country. So why am I telling you about this? Well, we took this website and we structured it, we made it consistent, and we were able to take the content out and put it into a data model. So this is now a representation of the treatment of cancer, the systemic treatment of cancer. I'm showing you part of the model here. We have more than this, a simplified version of it. But basically the idea here is that you're treating cancer with a combination of drugs in a complex schedule that's being repeated over time. And you have to know all these details to really understand what's happening. You also have to know the context in which you're treating cancer. And then on the right of this model, it's pretty important to know where these treatments came from. How did they come about? Were they compared to other treatments? Are they better or worse than other treatments? And putting that all together, we get to, at this point, over 100,000 concepts and 400,000 relationships. So pretty complex. And this doesn't actually capture everything, so we're still working on growing this. Portions of what we have are in the NCI thesaurus, which is the National Cancer Institute's thesaurus, where we capture basically everything relevant to cancer. And this is a teaser slide to say, if you are interested in this data, we actually have a lot more data points than what I just showed you. We have over 11, I think more than 12 million data points right now in various tables that represent the spectrum of the situation. And this is free for academic and non-commercial use. And I would be happy to talk to you about how to work on projects related to this. So the last thing I'm going to just say quickly about, has anybody heard of ACR Project Gene? I'm going to say, yes, you have. Do you want to say, have you worked with the data? Or you've heard of it?
Yeah.
They are awesome. Yeah, I'm glad you mentioned that. So if you haven't heard about it, check it out, because it's one of these pretty rare, hard to find, free and open access data sets that puts together the phenotype and the genotype. Rare, right? Like TCGA, which Dr. Zoon's going to talk about, is great for genotype, not so good for phenotype, right? And there are some data sets out there that are better for phenotype maybe, but not so good for genotype. So this really lets you start to ask questions of precision oncology. So this is like was said, it's a little blurry here, but Memorial Sloan Kettering is a big contributor. It came from Vanderbilt, also one of the founding sites, and basically there's sites in the US and internationally that contribute to Genie. This is just some overview numbers. There's over I gotta update this now, but this is 2 years old. There are about 150,000 patients in the gene data set. And a number of papers have come out, including a New England Journal and others using gene. So why am I mentioning it? Because it uses HEMOC, right? So basically we are expanding gene. It's called the BPC data model. So here the idea is we're expanding the phenotype characterization in gene to include information about treatment tied to the HEMOC concepts, and also information about outcomes. And Dr. Zin is going to talk much more about outcomes and recurrence in particular. Personalizing with that, I'm actually going to transition here. I'll come back up later and we'll talk more about our strategy.
Hi everyone, some of you know me. I'm Ejuzo. I'm a clinical bioinformatician, so I'll talk about my work with OPEX. So you guys are being trained as computational biologists, and my work actually is a representation of what a bioinformatician can do on the clinical side as a hospital setting. So I'm an engineer from training, and I did statistical modeling through my PhD, and during my postdoc, I became a bioinformatician, and I was trained at Brown. So I'm basically I joined Brown Pathology in 2016, and I founded Clinical Bioinformatics Center at Hospital. And I'm based at molecular pathology, and I am the founding director of Clinical Bioinformatics there, and also I'm the associate director of CCIDS. I'm also editor-in-chief at JMI. So what do we do on the clinical side? So day-to-day, basically, we receive patient samples, their history tumors, and to our lab, and we sequence them. And simply, what we do is we detect mutations in those. We look for actually mutations which can be biomarkers. Those mutations, some of them can be treated, some of them can be diagnosed with biomarkers, some of them can be diagnosed with biomarkers. And we detect those and then report it back to oncologists and hematologists like Warner. So they treat patients. So I have a team of people on the clinical bioinformatics side. So we basically develop pipelines and analysis pipelines. And I don't tell you what pipeline is, you all know. And day to day, we basically analyze data because our sequences are constantly running samples. We also build genomic databases, like the information. The databases are everything for us because we have to interpret the data. We have to find the clinical significance of the mutations, like what they mean for the patients. And that's why we keep everything in the database. So we build databases. And then we also build custom-made analysis tools. Some of them are for pathologists working with us, some of them are for us in-house, some of them are online available, which I will talk about. So this is the workflow basically. We have the multiple pipelines we use because we have to be set. We have to make sure that we are not missing any mutations. So you cannot have just one pipeline. You have the secondary pipeline to make sure that you are detecting every mutation. So on the clinical side, I mean, in research side, you can do undo. We can do undo as well, but they actually affects turnaround. So sensitivity is so important. So that's why we run two pipelines, and then everything goes into the databases, and then we have a variant interpretation process, which takes like 24 hours, and multiple people work on it. And on the clinical side, we don't right now at the Divine Hospital, we don't do, for example, whole exome sequencing. There are pros and cons in terms of getting whole exome whole genome sequencing. On the research side, because you want to learn everything, you want to see everything. You do a whole transcriptome, you do a whole exome, you do a whole genome. But when you do whole exome sequencing, for example, especially for the somatic mutation detection, because we don't know every single mutation there's clinical significance, we only target the mutations or the regions we have information. So we call those hotspots. So right now we have 2 live panels. The third one is coming in, and we will test the fourth one. So of course, I mean, basically my clinical work affected my research too, and we over the years we developed some tools, and I decided to make one of them web-based tools so that everyone can use it. So there are some online databases for various information. Some of you might know those bioinformatician is one of them. It's the user interface of the CCJ datasets. There's also Cosmic, which is like my best friend because it's the database of all somatic variants detected anywhere. There's ClinVar, which is for the clinical significance of variants. You can find any variant reported in that database. There is Nomex. Some of you probably use this. It's mostly for the population, the frequencies and the germline. And we have to, of course, distinguish somatic from germline. And then there's OncoKB. This is actually a new-created resource where you see actually important cancer genes and the hotspots. And then there's, of course, you see the genome browser. Actually, most of you use this. So this is open all every day on my computer, in addition to IGV. Okay, so we were thinking that when we do variant interpretations, one of the pathologists said, "Well, I get some of the information in the data as an Excel file, but I keep going back and forth to this on my database." I just told you guys about. And in 2020, we actually built a web-based tool. We call it WiseTech. So recently, two of my students, Ben, he's a medical student, but he's also a computational biologist, and Nitin Sirkumar, he's a undergraduate student. They worked on an updated version of WiseTech, and WiseTech was published in 2020. We are hoping to publish the updated version. So what WiseTech does, basically, you enter the variant, and then it gives you information on Cosmic, ClinVar, CCIDS portal, you see genome browser, and then clinical trial. So you see actually there are clinical trials for those mutations or for the variant you are seeing to targeting the variant, any of the drugs available. So you basically see everything in front of you. This is a simple tool to just save time for the pathologists or the variant scientists working on the clinical significance of variants every single day. So another aspect of my work has been gene variation detection algorithms. So insertion deletions are problematic, especially on the clinical side. We need to basically report the exact insertion or deletion, and some of them are complex. You might see an assembly and then there's an insertion next to it, and you might see something else, and the whole thing is in the back. So we actually developed an algorithm. We called it Indel Finder. George Sullerson, he used to be a research assistant at my lab. He's now a PhD student at biology. He basically is actually working with someone, you know. He works with Jeff, and he basically worked on this, and he has written this in R. And this is a very sensitive program, and we benchmarked it with the non-Indel detection programs, and it was pretty sensitive. We used this for a while in the hospital setting, but then since I started problems with R, just in the discussion with me. The programming languages we can use on the clinical side are very limited, so because of the security issues. So anyways, okay. So this is my clinical work. So this is what I do. But then I have also a research group, and I mentor students. Right now I have 12 mentees in my group. Jesse, sitting over there, is my PhD student. So we work on machine learning-based predictive models. As I mentioned, I am a mathematical modeler from training. So I kind of transitioned that work into machine learning, and I've been really enjoying this work. So we have other group members work on machine learning. Nitin actually, because he finished the work on WiseTech, he also started working on another machine learning-based model. So Carmine, who is the master's student, Jesse, and Dennis, is also a graduate of computational biology. She was a PhD student. Now she's a second year medical student. They all work on machine learning-based models to predict cancer recurrence. So Jesse has been working on glioma, Dennis has been working on breast cancer, and Carmine has been working on lung cancer. And I'll just give you some some information about Jesse's work. So we basically asked the question if we can use machine learning models to predict glioma recurrence and use genomic data from patients, specifically somatic data, and plus clinical data. So if you look at the literature, most of the machine learning models to predict cancer recurrence are they use EHR data. So you can actually capture the recurrence, but it's limited. So there is also biology behind it. So I wondered if we use the somatic data, the sequencing and methylation data, if we can actually do better predictions. So the data says we use TCGA data. Yes, it has problems. It's very noisy. Just spent I don't know how many weeks cleaning the data. And that clinical data has so much missingness. And I've been actually really collaborating with Dr. Warner on the project, and we kept showing him different clinical features, and it's really it was. I think it took just to clean the data than building the model. And that the validation data set, which we were very lucky because it's hard to find patient genomic data like separately all together with the clinical data. And this is a blast data set, it's completely independent from the ECGA data, and we used it for the validation of the model. So this is Jesse's model. So it's a neural network model with an attention layer. I'm not going to get into all the details of this. So specifically, we predicted early versus late recurrence. So from our conversations with Dr. Warner and other clinicians, we basically decided that, not decided, but it's obviously cancer comes back at some point. Maybe tomorrow, maybe in a month, in a year, 10 years, it comes back. So originally, my approach was we should predict recurrence versus no recurrence, but there's no such a thing. So we basically classified the data in terms of early recurrence and late recurrence, which is the cutoff is actually 1 year for glioma, and then we built the model. So this is the TCGA data results. We benchmarked it with traditional machine learning models. So, and the model, just say to name it Lunar, by the way, I forgot. It's a very nice name. I really like the name. And Lunar model, the AU series actually you see it here is 84.9, and it actually performs best compared to the traditional machine learning models, and we got really comparable results with the validation model. This, of course, after hyperparameter tuning, feature selection. This is months of work. So, but we will work on it. So for this model, we used only RNA sequencing, DNA sequencing, and clinical data, and we will hopefully implement methylation data, but this is getting ready to submit this as a manuscript. So, okay. So the other, yeah,
like mutational data, do you think you'll insert?
Sorry?
There's mutational data?
We have mutation data.
yeah,
Yes, okay.
mutational data. It's via gene expression mutational data,
Okay.
and then some methylation data in TCGA, but we didn't use it. Yeah.
Also, the results are now 91 for ARSC and 80 for APS.
What did you do, Jesse?
It's just we used the whole one. Just I don't want you guys thinking I have 64% ARSC.
That's not like.
Did you change it? This is
I think the word in the folder must just be wrong or something.
I'm using the incorrect figure.
Yeah, but it's totally my fault, I'm sure.
Sorry. But okay, it performed the best basically compared to the traditional machine learning models. Okay, we'll talk about it tomorrow.
I'm so sorry.
No, it's okay. Okay. The other aspect of my work is actually on protein-protein interaction. So this is a collaboration with Zoom, who happens to be my husband. So it makes our collaboration easier, I guess, because we see each other. So right, we should see each other. And he's also a CCIDS faculty, and I'm sure some of you have seen him. We have been studying protein-protein interaction. So I'm not gonna get into what is the network of protein-protein interaction. This slide is mostly for not computational biology people. So as you know, protein-protein interactions are dynamic. So they change from normal to disease state. This is not recognized. People have been working on this for a long time. And what they have seen, one of the people who worked on it is Dr. Barvasi from Northeastern. I went to Northeastern, so I've seen him. He has great work into not just protein interactions, but he also works on social interactions. He has a very nice book if you want to check it out. So he has shown that protein interactions are unique in different disorders like ophthalmological disease, cardiovascular, immune disorders, and it's the same for the cancer too, in different cancer types. So what iPad Group did, they basically built a tool and called it Proteinarium. So this is actually different than traditional protein injection tools. So normally you would have a differential gene expression experiments, and then you will take the differential expressed genes, and now you will map it to the interactome, and then you will see actually the protein injections in it. But what you are doing here, if you are working on a cohort of patients, you are basically ignoring the fact that protein injections are unique in every patient. So what they did was they basically mapped the protein infections in each and every case, and then clustered them to find a consensus network. Okay? This is what they are talking about. So during COVID, we were having a staycation. I was talking to what I talked about, right? And they were actually applying this method to other disorders. And I said, why don't we just work on cancer? Why don't we apply this to cancer? And we took our laptops and then we ran the first analysis in our backyard. It gave actually good results. So we decided to study other cancer subtypes. So as I'm in the pathology department, one of my surgical pathology fellows, Sean Hicking, he's an assistant professor now at NYU. He came to me, he said, we should study MMR deficient breast cancer. This really low actual prevalence. But he said, well, it's considering the prevalence of breast cancer is high. So we should study it. So we basically ran Proteinarium using a cohort of patients with MMR deficient and MMR intake data. So just to give you an idea, mismatch repair proteins are important, and we use them in the clinical practice for example, Lynch syndrome diagnosis, and they are not really, we don't really check MMR deficiency in breast cancer, but the MMR deficient breast cancer is 2 to 3%. And the mechanism is not known. So when we looked at it, I'm not going to get into all the details. We found that the MMR deficient patients share this consensus network, which is heavily histone proteins. So basically, my previous slide was showing, but I didn't mention it. The most connected protein in a protein network is a potential drug target, because in theory, if you target the protein, you might be able to take down the whole network, which might be tumor, right? But the MMR intake consensus network basically didn't show us the same results. So we published this last year, and we made some suggestions in terms of the MMR deficient breast cancer treatment. Of course, we need to do more like experimental patients on it. So the other thing we did actually, we studied 10 other, actually, breast cancer was one of them, 10 other cancer types. And we uploaded all the data on a vector. We called it upload protein infections in cancer. And over time, people worked on this. Undergraduate students, graduate students, medical students, and pathology residents and fellows. And this is I basically asked one of the students to make this chart. This shows actually, the small one shows the protein infection maps in different cancer subtypes, and then when you overlap them, as you can see, some of them overlap, but some of them don't. So I call this actually ugly picture of cancer. So if it is online, you can check it out, and it's an interactive vector if you're interested in protein interactions. So when you check it, let's say you pick a, like you will see different cancer types. You pick one of the cancers, for example, breast cancer. And then if you click on one of the proteins, it will basically show you information from human protein atlas, HNC, G profiler, CLU, and that SIGWare portal. It will give you the survival rates and all this information. And the red proteins are the ones with existing drug. They are existing drug targets basically. So this is the QR code. And these are my collaborators and my lab members. Thank you very much.
So we already heard a lot of smart things. I'll just remind you two things. You've heard that data is the oil. I wonder if data is the new snake oil. And the other thing I would say that all models are great, some are useful. And why am I saying those things? Those are basically the direction I follow. Neither a clinician nor a bioinformatician. I started as a structural biologist. I've been interested in data. I joke that I would rather use someone else's data than generate my own data that nobody may ever use. So those were the principles under which I met Dr. Warner back at Vanderbilt. I was finishing a postdoc in immunology. Wasn't going exactly as I wanted it to. So I have been debating about what my role is. But my role is, I am the coordinator for the CCIDS. If you are interested to know more, or if you're interested to debate more, or you want to be involved in the CCIDS, I would be your contact. As I said, I finished a PhD in structural biology back at Vanderbilt, did a postdoc. I have been interested in various aspects of the data, and that includes the ethics, the regulation. Those are my day-to-day job. So if any of this interests along with the CCIDS's goal and principles, then I would be your person to reach to.
So we're going to just talk a little bit about our vision. Our center has been in operation for about 18 months, just give or take. Right? I believe these principles still hold, but we are evolving as we grow. So I'm not going to show you the 5 again, but I'm going to go through them. So content standardization. Hopefully you've kind of got a sense of what I'm talking about here, which is that if we don't all speak the same language or have a model or set of models that we can agree upon, we're going to have too many different results that don't match up in any way. This is a good place for that XKCD cartoon, which I should add in the future. But we work a lot on standards development. And as you can even see here, there's already too many standard setters in this space. But they all each have a role in certain aspects of the cancer conundrum. And so, unlike wall plugs, where there's one and only one for the United States, there are many solutions here. But we need to get people to work together, and we are thought leaders in this space. We work pretty closely lately with the US CDI, which is the national body governed by the Office of the National Coordinator, ONC, which actually tells the vendors of the electronic health records what they need to have in those records. And we are pushing on those vendors to make the ecosystem more interoperable. So that's one item we'll work on. Number 2, clinical integration. So I think you got the flavor of this, right? There's different aspects. I'm going to show you this next slide, next couple slides, some more about this. Integrating and improving the clinician and the bioinformatician workflows. And this is not just here in our clinical domain, but also around the state of Rhode Island, potentially beyond. So here, I'm going to just show you some slides that I presented almost 7 years ago at the National Academies. Okay, just to give you a little timestamp here. So does anybody know what that is? So blurry, but what is, does anybody know what that is? Maybe definitely come to this in your lives. That's a fax, right? Something that That was the state of the art clinical report for genomics. You can barely see it, but there's a list of findings that have a potential intervention. Well, we worked with this company called Foundation Medicine back then to get a PDF, right? It's a slight improvement on a fax. It's full color. It's much clearer. It's computable, maybe. You can pull things out of that. And we also worked with them to create a standard representation in XML, basically, that we could put into our medical record. Now, this is getting to be an ancient screenshot here, but this is Vanderbilt's custom electronic health record. And we had the mutations from this report right in front of the clinician. Unfortunately, what I don't have here is the arrow that goes back to the beginning, because here in 2024, we're getting faxes over at Rhode Island Hospital. We're back to the left. So, you know, we made it to the right at Vanderbilt, and there's other places that have, but this is by no means consistent around the country. This slide I also showed, I want to maybe move this up in future versions of this presentation, because I think it's important to, let's see the lower right here. It's a, you know, we really represent these two sides of the coin here, which are the clinician and the laboratory. And we have shared tasks and we have our own tasks. And I'm just going to quickly kind of show you what I'm talking about here. So there's a process here. The process begins with ordering a test. So you've got that tissue that Dr. Rizwan talked about. What are you going to do with it? What kind of tests are you going to run on that tissue? That's a shared decision. Right? And then you actually do the process. That's where kind of the pipelines get in, the measurement, right? Well, from those measurements, you make an observation. What have I just seen? This is also getting from that data to the information to the knowledge. The knowledge, what does it mean? So I observe something, but what does it mean? Is it a prognostic finding? Is this a cancer that's going to be treatable? And if so, what are the treatment options? That's a shared task. Back to the clinician, decision making with the patient, right? It really should be it's shared decision making. Always with the patient at the center. We're going to take all this information and make a decision and potentially make an action, a treatment decision, or a decision not to treat. And I'm going to do this over and over again. So that's kind of the nature of this. You know, as Dr. Rizwan said, most cancers recur, not all, but most. How are we going to monitor that? How are we going to detect that resistance is coming about, and what are we going to do about that? So it's kind of the whole process here, the circular process. This process, even though it maybe looks kind of smooth here, it's pretty rough around the edges. Going from step to step takes a lot of manual labor. Information gets lost in these cracks, these gaps. And so there's a lot we can do to improve and model for others. All right, information extraction. So two ideas here. One is we want to continue to develop best-in-class natural language processing solutions, cancer phenotyping at scale. So that would be through DeepFeed, one project. Just this week, got a notice of award for R01, where we're going to work with who we work with? Brigham, right? I think it's here. We're going to work with radiation oncologists up in Brigham Women's to detect immune-related adverse events. So that's another key task. It's really important because immunotherapy is very common these days and has a lot of side effects. Idea number two is that we want to provide a local service. We did this at Vanderbilt, and as I was saying earlier, this is hard here. Getting clinical data here is hard, more difficult than anticipated, but we're still working on it. And we want to engage with you all to come up with questions that might benefit from clinical data here and how we can go about that. We are, by the way, hopefully you've heard this, we are working on building biospecimen repositories at the hospital, because what you all really want, right, is both sides of this, the genomics, the biospecimen, and the clinical data. Number four, we didn't really talk about this aspect here too much, but dissemination into new technology. The vision here is to really identify and promote new innovations that are going to be into the clinic in the next 5 to 10 years. Something that was completely absent from this talk was single-cell sequencing, spatial transcriptomics, what else? I don't know, proteomics. There's a lot of really interesting technologies out there, but they're really far from the clinic. We want to figure out which ones of those are actually going to be coming into the clinic and help promote that translation process. Getting technologies that you all work on into the clinic is a really long, arduous process. It can take decades. We used the example earlier of CRISPR, right? That's maybe one of the fastest. You'll hear that there's a new CRISPR. The first CRISPR therapy was just approved by the FDA. I think it took about 10 years from sort of the science coming to the treatment being available for sickle cell anemia and thalassemia. That's really fast. A lot of new technologies take a lot longer than that to get into the clinic. But we want to play our part in making that happen. And then the last part is scholarship and education. So part of that is just coming and talking to you. Another thing that we basically established is a seminar series where we really are trying to serve Rhode Island and the broader New England community to bring in people to talk about clinical cancer informatics and data science and their work. We're also working on training programs for Spectrum. We're actually right now we're submitting one to train high school students in cancer and data science. We kind of want to cover the spectrum from high school all the way up to postdoctoral level. So just a word about our seminars, and one takeaway hopefully is that you'll come to our seminars. We've actually had a lot of good speakers, a lot of interesting folks. Say Mick, you can see from just look at the degrees, it's been like a mix of clinicians and scientists, and of course people were sitting in both domains. What else can we say about this slide? Just that we've speakers, and we have some that are upcoming. So I'm actually really excited about Sean coming. So we mentioned AACR Genie earlier. So Sean is the executive director of Genie. He's coming in. Yeah, that's 2 weeks. 2 weeks. And we are holding these seminars over in the medical school. So that's right, CSR 280.
Oh, yeah, those are all med school. The reason why the charge I told him is because that remains our backup. We have been widely doing it, but still remains our.
Yeah. So Dr. Sweeney is going to be here. We have free lunch, by the way. And in October, we have Dr. Sonia Reed from Vanderbilt. So she's the health disparities researcher in breast cancer. We have, I think, well known to this group is Dr. Ben Rafael coming in November to talk about his work in bioinformatics. And then in December, we have, we call him Joe. So he recently moved from MSK up to MGH. And he's their director of pathology informatics. So he's going to talk about that. That's the last slide we have. So we can open up the questions if we want to come back up here. I think we have a few minutes here. And just a reminder how to get in touch with those things.
Research tools. The United States has no socialized medicine, and there's so many different EHR record tools used. What do you think are some additional anticipated challenges regarding the integration of these tools and having
Easy answer. No, yeah, so this is exactly, the issue around standards, right? So there's a couple of philosophies around here, right? One philosophy is just demonstrate best practices and then they will just start doing it. Another philosophy is regulate. There's both philosophies, right? But so we tried regulation in this country. We had something called meaningful use, which happened about a decade ago. At first, people went along with it and they're like, sure, we'll standardize gender, race, ethnicity, some easy stuff. But then as it started getting more complicated, the vendors started chafing, right? They're like, oh, we'd like to do medications our way, right? Or diagnoses, we have our own thing. And so that kind of fell out of favor. So we're back to that sort of demonstrate best practices area. One way to do this is to get patients the demand to change, right? So I didn't talk about that in this talk, but I work with a lot of patient advocates as part of the what's it called? Open Notes. It's a movement that basically said, okay, patients should be able to see their own notes, and that will affect some change. They should be able. There's something called the 21st Century Cures Act. Right? That's regulation, actually, which said patients should be able to download parts or all of the record in some way. They shouldn't have to be handed a stack of printed papers, right? So slowly but surely we're moving in the direction. So the latest thing is this USCDI Cancer Plus, USCDI Plus Cancer. We have some folks there. We're building it around some specific use cases. The most interesting one is clinical trials matching, which Dr. Reason talked about a little bit, right? Like clinical trials that have, you've got a common situation where you've got a patient who might want to know what their trial options are. And you actually need a lot of structured variables to get to that match. So that's what we're working on right now. But it remains to be seen. I have to say, even countries that have universal healthcare somehow can't get around the issue of multiple vendors, EHR vendors. And Australia is a great example. They had control of their system pretty good, but then they just recently decided to go to Epic, and it's like they're back to square one. So
I'm glad that you brought it because this is where the real world conflicts with all the great imaginations. Some of the things that can make a big difference are geographical and local factors. But those factors also require that you have to have detailed information about where you live, what race you belong, and what are the other details that can be temporarily over the period can be tracked. But as you can imagine, they also open the challenge of the privacy and other factors. So I think once you are aware of those limitations, not only that helps as you grow over your career to think how to overcome them, but that also gives you a skepticism as to whatever data you come across, how much of that is appropriate to be used for what purpose. I think this is where the scholarship part of the center comes in. We provide that kind of thinking. Sit down and think how, what is the limit and what is the value utility of what we have.
can I ask you all a question? Oh yeah, go ahead.
How about integrating omics, genomics, in terms of single cell discovery with these medical records? Sounds like there's already a mess in the medical records without.
So that's what Genie
So
is. Genie has been doing that.
Genie has taken a bite out of that. It's the panels, right? So Genie has big numbers, multiple sites, kind of gets around that whole bias thing around a single site study, for example. But it's the panels, so it doesn't necessarily see what's in the whole exome, what's in the non-coding regions, right? So there might be something important that Genie doesn't even pick up on, but it's a start.
When you say the panels and the panels are
There are like 500 or 600 genes typically, and 19 regions. So, oh, smart.
This
Well,
is, yeah.
Well, like MSK and Dana-Farber,
Yes,
they're about 1 million base pairs,
Yeah.
more or less, out of the 3 billion. So,
we focus on the population.
yeah.
Yeah, the parts of the genome we know the clinical significance. Because otherwise it takes months to create the data, weeks to months. Create the data and come up with the clinical significance.
I mean, UK Biobank or other biobanks, large scale data sets with phenotyping.
Germline, right? Yeah. So I think that's so the perfect data set that has somatic and germline and clinical doesn't exist, right? And that's,
I guess
so
We had,
we
it was
think
maybe
about how
great
what-
for JC.
we think about what it would take to create it, but I will say, I'm going to say a number here, and like I don't know, 50 million is the number. That's what it took for Genie to extensively curate 15,000 of those 150,000 cases. $50 million. So that's a big part of this problem.
And since we have been both part of the Genie, the challenges that are sometimes insurmountable, primarily because you have the gene data, which is huge to begin with. So you have to bin what you can. And then you have to then go identify the patients and then go a comprehensive record of their EHR, which at this point is not electronic. It's not like you just download it and merge these two files. Yeah. So oftentimes you have PDF records, you have image data that then has to be humanly read and entered through something like RedCap, which is in a glorified Google form, where somebody manually has to read this, enter this, and then you create data. So whatever little the Genie has, it's very valuable, and it sets a benchmark that can be done. But even that was done with a very limited, purposely it was done with a limited sort of curation expectation. So
this is where tools like DeepFeed could come in and solve this problem. So, another number here, 8 hours, that's about how long it takes to
One
curate
case.
one
Normal
case, a normal case, that's right. And that's for someone who's an expert curator, like PhD level, highly trained or clinician, who knows what they're looking for in the medical record. That's what it takes them. So very time
And
consuming.
that's for the lung
That's
cancer and
really
prostate
manual.
cancer. For the breast cancer, where patients have a much longer record,
Mhm.
you then extend this to a day and a half. And you begin to wonder how much of this you actually want to wait, because these patients go 6 monthly scans. Do you really want to read all of them? When you lose that, you don't know where in the future that might be useful. So those kind of balances.
So that's where like the aspiration of a tool like DeepFeed could really, come in. But it's difficult, it's hard work, yeah. Did you have a question there? No, okay. I did have a question for all of you. How many of you do anything cancer related, even in the, tangentially, fully, anybody do cancer related work? All right, not as many hands, okay. And for the rest of you, like, what do you, like, what areas do you focus on? Other diseases, normal health, non-human, like what is the general?
Associations.
Not necessarily in people,
But
but like in.
and fruit flies are the most important organism.
I got a bunch of my customers, they think they're from here. It's their season.
I think
Contributed with your thinking.
CCB did a lot of population genetics and it's less than story to just genotype.
So like healthy population, or is it like
Like
the UK
there's-
biobank?
Yeah, well, yeah, I mean, there's some like people who do UK biobank, like just like general phenotype, not necessarily disease phenotype.
How about all of us that want to work with that? You work with all of us?
Speak up.
So yeah, do you want to say more? Like, what have you done with all of us?
Right now, mostly clean all of their plain files, but also do a lot of stuff like the survey and our clean files.
Link files. I spent my postdoc thing. Yeah.
So we actually, so there's, so the thing with all of us is it's not been around long enough to really collect those people. Usually people sign up when they're healthy, they don't necessarily have cancer right when they sign up, right? But we actually have a project with UCMB where we've looked at the subset of people with cancer and all of this, and it's about 100. I don't know, it's a substantial number, but we just started to look at that. You don't necessarily have their, so again, like there's no perfect data set that has somatic and the germline and the clinical. So all of us has the germline, right? It has a good proportion and it has pretty nice clinical phenotyping. But it doesn't have its
No perfect data set either.
not yet. You
Especially
gotta build
when
it, right?
The issue, yeah,
It's
especially
$100
on.
million.
Yeah.
All right,
Yes,
we'll
yeah.
work on it.
Okay.
All right, well, thanks so much, everybody. We're around. We're happy to send our contact info out for slides. So share them. Thanks again for